Hello. Today we are going to talk about single cell sequencing. This is a follow up from our last discussion of a month back, and we spoke about transcriptomics and genomic variations. So, single cell sequencing is one step further. Like transcriptomic variations in general, when we discussed it, was mainly focused on bulk level sequencing at a whole organ, tissue, or multiple cells together, but single cell sequencing involves transcriptomic profiling at a single cellular level. So, why do we want to study single cells? Well, and especially for complex human organs that consist multiple cell types, it's hard to identify individual cell type level signatures when you do bulk sequencing. So one of the ways is to drill down to individual cell level. That helps to look at the expression profile of rare cell types, and also identify some novel cell types that are not expected at that time of their sample collection. Also, it helps in characterizing the transition between the cellular stages. It helps to look at the dynamics of transcriptional changes during development or aging or disease process. In addition to looking at single cell transcriptomics, even at a single cell level, you can look at chromatin assemblies by ATAC-seq. You can look at mutations and CNVs by single cell DNA sequencing, methylation, as well as single nuclear level sequencing. While the technology has advanced rapidly, it brings in new challenges. One of the challenges is the amount of RNA that comes from individual cells. There are cells that are large enough in size to give good quality RNA, say 100 grams, that is needed for regular sequencing protocol. However, there are other cell types that have limited amount of RNA, and in those cases you need to do some kind of amplification. So that's a technological challenge. Secondly, think of the condition if all the cells have all the genes expressed at the same time, then there won't be a lot of differences between them. So that's why when you look at single cell level, you may have the whole genomic library of 20 to 30,000 genes, but not all the genes will be expressed in all the cells. So you cannot just assess all the genes in one cell. And that leads to analytical challenges of getting multiple rows of all zeros or very low expression. So let's just walk through what's a simple workflow of single cell sequencing. Well, similar to bulk sequencing, you collect a tissue part here. So for sampling purposes, just talk. This is a liver. Snip out a part of it, and then using enzymatic dissociation, separate them into individual cells. Dissociation protocols have advanced. Initially, it would be by one or two enzymes like dispase and collagenase. However, now there are pre-customized enzyme cocktails that are available from the manufacturers directly. You can throw the cells in those and they will get separated, and then individual cells can be isolated using either fluorescence sorting or FACS, or magnetic sorting or MAGS, or any other protocol. Once we get the individual cells isolated, and now the protocol proceeds the same way as in bulk sequencing. However, instead of one sample being one unit here, each individual cell brings out its own RNA and is sequenced. So once you have the cells separated, then RNA isolated from it, then cDNA libraries are generated. They are either amplified or they are even fragmented into parts, tagged at the end, and then each of those libraries are sequenced. And once you get the expression profiling, you can cluster them and identify the individual cell types. That's in short the whole process of single cell sequencing. Once you identify cell types, then you can do multiple things. You can do differential expression within each cell type, or you can also look for I guess, longitudinal studies, how over time the cellular composition changes. So the ability to measure single cells has been feasible for last few years, starting in 2009 and 10. At that time, it was more or less manual isolation of cells, isolating individual RNA, needed a lot of effort and for very minimal output of RNA, but that was the start. Gradually, things moved from single tube to multiplexing, and then to microfluidics. A lot of these microfluidics approaches are still working. Like Fluidigm is one of the examples. Then gradually, as the technology evolved, robotics were involved, and then around 2014 and 15, droplet based approaches were developed both at institutional level as well as commercial level. And these technological improvements have led to a exponential increase in the number of cells that can be sequenced. So at one point, there in 2009, they would sequence only one or two cells, and now around by platforms like 10x Genomics or Dropseq, you can sequence thousands of cells taken together. And then each of the cells are barcoded, and then individual counts are generated from them. So the one of the things that you have to remember, and I think we discussed that during the bulk sequencing, how from 2008 to 2015 the RNA-seq data generation saw an exponential jump, and I think right now it's the same thing happening for single cell sequencing from say 2017-18 to 2021. You will see a lot more single cell sequencing data sets in resources like GEO. So once you're ready to do a single cell sequencing, the big question is you have to choose how many cells you want. That depends on what is the goal. If the goal is to identify or characterize some unknown cellular types, then you may want to go to cover as many cells as possible within a given budget, and that would need the depth of sequencing will be less. It is a shallow sequencing cover a lot of cells. However, if the goal is to identify or characterize some rare cells, then it's advisable to go deep level sequencing only a few of those rare cells. So that the choice is uh, dictated by the question as part of experimental planning as well as the budget. So one of the things that single cell sequencing leads to is because each individual subject may form a batch, then there are that leads to technical batch effects. And there have been ways to fix those. We will touch base briefly on some of the approaches being used. And of course, big cost is finances. A typical single cell sequencing run on a platform like a droplet based platform like Chromium costs approximately $2,000 for one sample, leading to say 100,000 cells. Contrast that to a bulk sequencing cost of one sample around $300 to $400. So there are resources that help you decide how many cells you want to use, what kind of UMIs you want to use, and what are the controls you want to use. And one of the great resources is the New York Genome Center's Satija Lab, and they have tools that help you identify the number of cells that are good for the question being asked. So let's talk about the molecular pipeline for a single cell sequencing project. So the typical flow goes from get the tissue piece, then dissociate using enzymatic mixture, and then use either a flow sort or magnetic sort to separate the cells in individual chambers or components, then lyse and get the RNA from each individual cell, do the RT, tag them into cDNA libraries, and then mix all the cDNA fragments together and do individual sequencing. So what are the challenges? One of the challenges is how easy or how hard it is to separate the cells. If it's something like blood, then the cells are not as sticky. You can separate them much quicker or easier. However, if it's a hard tissue, then there's need a strong enzymatic co- cocktail or mixture to separate them properly. Even after doing that, there is always possibility that one or two cells sticking together and causing a doublet. So those things, those are identified at the analytical level by doing some QC of the data. And also the cellular separation and RT protocols differ by how you are use I guess separating the cells, whether it's by flow cytometry or magnetic or any other approach. So there are different cell separation approaches. There are some that are for low throughput, for fewer cells, but they're more specific. Something like fluidic platforms such as Fluidigm. They are usually an integrated platform. So you take the cells, the Fluidigm will separate and throw them in a well and then bring out up to the RNA level. Usually is a limitation in number of cells that can go in through that. It's around only 10% of all the cells that are captured. So you at most you may end up with 100 to 200 cells from individual subject. And because of the sizes of the chip, it also limits the number of cells then that can go through. So these are highly specific. You know what cells you are getting are the cells that you are looking for, and they need a lower volume, so you save reagents and save money. However, in long term, you have to decide that whether you want to have a limited coverage, only a subset of cells examined, or you are looking for exploring the cellular level transcriptomic topography, then you may want to query a lot more cells. And then using a microfluidic based method gets more and more expensive, and that's where you want to use some droplet based approach. So in this approach, you basically take each individual cell into a droplet along with a bead. The bead is loaded with a tag, and then that also bead contains the enzymatic composition for generating the libraries as well. So each cell is separated and created into one droplet, and then within that droplet the RNA isolated and the cDNA libraries are generated, and then each droplet is further sequenced. There are multiple droplet based approaches. One of them is called DropSeq, which is it's a highly customizable platform, and these are primarily created and used in individual academic laboratories. And then there is a commercial platform called 10x Chromium, which has a lot of the kits and reagents that come directly from the manufacturer, and that helps in reducing the time and effort, and also it increases some cost because it's just create things are pre-made, so you pay money upfront. In terms of cost comparison between the high throughput and low throughput approaches, something like dropseek, it cost say up to 5 cents per cell. However, when you look at the number of cells it can query up to, which may be up to 200 to 400,000, then that jumps a lot more than when you are looking at the fluid ion based approaches where it's only a few cells at a time. So let's just review briefly the protocol and how these individual approaches work, starting with the fluid ion or microfluidics based method. So it works on a chip. This is a chip, and now if within each of these, in this chip, these are the wells. Now within each well there are these tiny fluidic microfluidic chambers through which the cells pass. So you take the cells, then load, capture, separate them using the chip, the microfluidics tubes, and then lyse them, amplify them because there will be very minimal amount of RNA, then to do libraries generation, preparation, and then sequence, basically analyze it. So all these things, these steps starting from capture cells to imaging and barcoding takes place in the instrument. And then once the libraries are generated, then it goes back to the regular any sequencer, and then the data is analyzed on a computer. So you can think about, again, comparing to what we already know about bulk sequencing here, and from each individual subject, you're, instead of getting one column that you get in bulk sequencing, you're getting hundreds of columns for each individual cell. Moving on to the droplet based methods like dropseq. So these use the custom designed microfluidic instruments. They create individual droplets, and the cells are then, each individual cell is contained into one droplet along with a barcode. This barcode and the bead in there consists of the reagents that helps in generating the libraries. Once the libraries are generated, then each of these droplets are coded, sequenced, and then that sequence data is analyzed into the numbers. At the end of the day, again, similar to the fluid ion based, fluid microfluidic based approaches, you have a matrix from one subject which has thousands of genes to few thousand cells, few thousand columns, and keep in mind that's from one subject. So if you have multiple subjects, then you get multiples of those columns. And the commercial product like Chromium, it's also droplet based method, but it comes with its own chip, pre-developed chip. You just have to barcode the beads and then throw in the sorted cells. They are added individual droplets in the chips themselves, and once these barcoded cDNAs are separated, it's amplified and libraries generated and sequenced. The analytical pipeline involves taking those barcoded sequences, aligning them using algorithm like STAR, then tag the genes, do QC, correct the sequences, then count and make the cell by gene matrix, and then tag each cell with its barcode, and then you get M by N matrix that can be imported to any analytical software. So here is a side-by-side comparison of the microfluidic approaches as well as droplet-based approaches, and there are some plate-based methods as well, where You separate the cells individual format. Those are also on the lower end in terms of number of cells. So both the microfluidics and place-based approaches have high gene coverage. They are cellular specific. However, droplet-based methods, since they cover and look at many cells up to thousands, the number of genes covered can vary depend on the specific cell type that is being used. What are the challenges? Well, while in the microfluidic device, you can see the individual cells. They are more expensive in long term because of fewer number of cells being generated and per cell costs being more. However, they do have high efficiency capture. But for droplet based methods, the positives you can count thousands of cells. The low cost of these, you don't need controls in there because the droplets use barcode to do the control, and higher cell coverage, whereas at the same time there are not many genes that can be covered. Sometimes it's maybe 8 to 5 to 8,000 genes is at most covered because they're covering way more number of cells. This is a comparison with showing what are the differences between the three most common approaches of single cell sequencing. Fluidigm can go up to 1,000 cells. Dropseq, you can sequence up to 150,000 cells depending on how you customize it. And Chromium, it says 1,700 cells, but things have improved now. You can even sequence 2 to 3,000 cells per run, and it can go even more depending on the cost you are willing to pay. So, and at the end of capture, you can get few hundred cells to few thousands, and then somewhere in between is the Chromium that is balancing both in terms of specificity and sensitivity as well as cost. This is how the coverage looks like. You can see there's not much difference between the Chromium sometimes is in between in the Fluidigm as well as Dropseq, but kind of makes a balance. However, I wanted to point out that there are not these three are not the only ones. There are other approaches as well. Something like Smart2, MarSeq, CellSeq, InDrop, and Sequel. They have similar approaches, and here it shows what platform they have. So either microfluidics place based or droplet based, or even there are some that have nanovel arrays in there. So basic step follows is identify the tissues or cell of interest, dissociate them, then capture at individual cell level. Those captures can be done at microfluidics using FACS or using micropipetting and run into Fluidigm or Chromium in Dropseq. So there are different steps associated with the method used. Well, in addition to deciding how many cells you want, the next step in terms of experimental designing is to choose which platform you may want. Again, if you are looking for specific cells and you think you can do good with few hundred cells, then the Fluidigm or C1 type method is good. That can cover way more cells than something like Dropseq here, which because of the large number of cells being covered, the average number of genes looked at is much less, while in C1 this is much higher. In terms of accuracy, you can see the Chromium is close to the top, and in terms of specific sensitivity, it is somewhere in the middle. So that's why we feel it's kind of good balance between the two ends. So one thing that keeps coming up when you are doing a gene single cell level analysis is this term called UMI. So UMI is a unique molecular identifier that gives a count of molecular level for each individual experiment, and these you can think of these molecules as corresponding to individual genes. Sometimes there are way more UMIs than the number of genes, and that situation, that's basically multiple UMIs go in mapping one particular gene, and you can think of it as in terms of a probe set of a microarray. So there are multiple probe sets associated with one particular gene, and UMIs help in reducing the noise and amplify due to the amplification, and also look for removal of duplicated sequence fragments. QC protocols are in place to identify these and remove them from final data set. The data analysis pipeline in general stays the same for whether you used low throughput or high throughput data. So starting with the analysis pipeline of Fluidigm, the steps are similar to bulk-seq, as do a QC, do the alignment, mapping the QC and quantify. Can the same tools like library preparation, mapping tools. So some most commonly used one is now STAR, and then look for QC by FastQC, and then quantify using either HTSeq or any other counting tool. It's typically one library per cell, so you may end up being hundreds of FastQ files. Need a relatively faster machine to process all that. You can then do the cell QC normalization and look for confounders. There are pre-existing pipelines that are already created for these. One of them is called Syncera. It is developed at Cincinnati Children's Medical Hospital. What Syncera does is it takes the expression, the count values, does the normalization, filtering, etc., and then using the algorithms of distance measurement, they identify clusters of cells, and then within those clusters they look for markers, basically genes that are different in each cluster compared to all the other cells, and then use those markers to identify the cell types. So the purpose is to identify major cell types based on the presence of markers and detection of additional markers for those cells, and that leads to identification of cell types, and many times leads to discovery of novel or unexpected cell types from those tissues. In addition to that, what can be done when you have the feature selection, basically the identifying the cell types, doing clustering. The next approach can be doing differential expression even within individual clusters. So if you are looking at a single cell level between two tissues, one from a diseased and one from an age match control, combine them together, and now when you have individual clusters, within each cluster you can separate the diseased and the control tissue and do a differential expression. When you look at high throughput data, like something like Dropseq, there are additional steps involved in there, something like corrections for UMIs and deduplication and quantifying them. They are all generally run in a single pipeline. The challenge here is, since you have thousands of cells, you have thousands of FASTQ files, you need tons of storage, and these are expensive to sequence. 20,000 cells up to 1 million reads. So the cost goes up, however the number of cost per cell goes down. And then there are customized analytical approaches for Dropseq depending on what you are querying and depending on the platform variability. One of the approaches used is called MAST. It uses linear modeling for gene expression data. and then has a GSE, a gene set enrichment type approach to identify clusters of and networks of genes and cells. MAST can be downloaded from GitHub, and one of the creators of MAST is Andrew McDavid, and you will have a class with him, I think, in a few weeks. So if you have any questions, you can reach out to him. He's in biostats in computational biology department in Saunders building. So even in DropSeq, once you have the genes features selected, then the clustering, you can always do differential expression within each cluster, or you can do dimension reduction like PCA. In Fluidigm data, it does not matter as much because you have only few hundred cells, but when you are talking about thousands of cells, something in DropSeq, dimensionality reduction by PCA definitely helps in better visualization and identifying of novel or unknown cell types. In terms of Chromium, this is a commercial based method. You still get thousands of FASTQ files. However, there is a commercial software already created for by the company. It's called Cell Ranger. And once you have the libraries, the Cell Ranger output does preliminary analysis and marker generation as well, and it also provides you the matrix that you can upload in other customizable software like Seurat, which is run on R. So the big question is, okay, you have the data, you have spent tons of money, created a single cell sequencing data set. What are you going to do about it? What's the use of it? So here, this is what you get at the end of all these runs. You have each dot here says a cell. You have transcriptomic profiling of that. You can use a set of genes from each of those and create gene networks. And using marker genes, you can use them to classify these cells into individual cell types. So one of the examples here is like these few cells that are underrepresented or rare state at this stage, for example, and you're not expecting them in, say, a developmental stage. Another use can be to look at the developmental trajectory. While an individual individual human being, their organ is, there is a specific age for them, but studies have shown that the development is not constant. All the cells don't develop at the same rate. There are variations, and that's where you can use something like Monocle to do identify developmental trajectory using something called pseudo timeline or pseudo temporal studies, and those help in looking at either normal development or disease progression or even treatment therapy outcomes. Now coming to actual parts of quantitative biology. You got all the data done, which looks like a matrix. You have genes 1 to 1000, you have cells 1 to few thousands, right? So you have this matrix, you can use some QC or filtering, do a normalization, and then next step is to do clustering, basically identifying groups of cells that may lead to cellular type identification. And for that, you choose something called variable genes. What are these genes? So these are the genes that are changing across the cells. Like you have thousands of genes, but a lot of those are not changing across cells. Then there is no variability, means there is no information that you can use to identify the differences between cell types. So we don't use them for clustering purposes. What we do is we look for genes that match a certain criteria, call them hypervariable genes, take them, use them for doing some kind of dimensionality reduction, either PCA or any other approach. And then using that information, cluster, group the cells, and find out the markers of those cells. And You can either query the database for the known markers of certain cell types, or you can look for differentially expressed genes, which are basically any genes that are different in a cluster compared to all the other cells, and also upregulated in those. And using those cell types, go to any pathway or cellular identification databases, look for what cells these genes are expressed in altogether, and then use that information to annotate the cells for to their subtype. And then other branch of analysis is can take the reduced data set, do some trajectory modeling, and then look for transcriptional dynamics throughout the process of, say, development or disease progression. Once you have the data, then you can play around, do anything. However, there are challenges that come with the data, and one of those we has discussed earlier as well as amplification bias due to the low amount of RNA that comes in. You do cDNA amplification. Then, of course, the RNA capture, because it depends on the cell size, cell type, may or may not cover all the genes. And then, of course, there are a lot of genes with no counts, because not all cells express all the genes. And also, sometimes the expression level is so low that you don't see any detection. But these are the challenges in terms of generating the data. Some laboratory level challenges are due to the cells sometimes undergo a lot of stress, and if they are fragile, they don't make it till the end. And even if you count, we have thousands of cells, quite a few of them may not be in a living condition to give you good quality RNA, and then that leads to a loss of the sample. Those are the challenges you need to overcome, but again, like any other biological experiment, that's part of the process. So, as we saw that from each individual subject, you can get thousands of cells. That's a ton of data. However, you just can't stop at one subject. Say I have 2000 cells from one donor, and this is the transcriptomic topography. That's not the way it usually happens. You'd run at least 2 or 3 subjects, do a single cell sequencing from each of those. You have thousands of cells from each of those. And that brings in another challenge, which is similar to a batch effect challenge in bulk sequencing. You have to combine individual donor cells from individual subjects or donors, or humans basically, and even animals. Well, animals, the genetic variability is as not as bad or not as high as humans, so their integration is much easier. In terms of human subjects, here's an example. These are single cellular data from 2 individuals from one of the project that I have worked on, and you can see if you just combine the data from two, there is a clear separation by the individual based on the color. However, there are approaches that help in removing these challenges. One of them is called canonical correlation analysis, or CCA. It identifies the matches between the two experiments or multiple experiments. Those are used as anchors, and then using those anchors, all the other cells are basically harmonized and brought together into a single object. And you see at the end of CCA, this is something that looks much better than the first image. Another approach that has been developed recently and used in updated Seurat version, that's called reciprocal PCA or RPCA. It also involves anchors between the two data sets. However, it uses reverse PCA. Is basically taking each data set and projecting into it the others PCA process, and then limits the anchors by the mutual neighborhood I guess, restrictions. Both these approaches can be used for integrating samples, which in terms can avoid, you can think of as like batch correction or sample matching. Think we'll stop at that, and I'll discuss starting quality control and filtering on Friday. So thank you for attention. We'll see you on Friday.